Hello, and thank you for joining Cure Alzheimer's Fund for this webinar featuring Dr. Vince Groppi discussing how venture capital and philanthropy accelerate drug development in Alzheimer's disease and other diseases. My name is Meg Smith, and I am the Executive Vice President for Research Management here at Cure Alzheimer's Fund. Our event today is possible thanks to the leadership of our event co-chairs and host committee. Jackie Morby and Henry McCants applied the keen insight and expertise developed in their groundbreaking VC careers, Jackie at TA Associates and Henry at Greylock Ventures, first to found and now as board members to shape Cure Alzheimer's Fund. Our host committee members, Sam Brash, Jeff Chambers, David Dolby, and Matt Kelleher, are active venture capitalists, and we are grateful for their ongoing support of CureAlz and our VC-inspired approach to accelerating benefit for Alzheimer's patients and their families. Today, we will get to hear Dr. Vince Groppi share his experiences and perspective as a successful drug development expert who has bridged academics, industry, and startups, and then hear his answers to your questions. You may type questions into the Q&A function in Zoom at any time, and I will pose them to Dr. Groppi after his presentation. But before I introduce him, please allow me to take a minute to introduce Cure Alzheimer's Fund. The founders of CureAlz drew from their VC backgrounds to shape our organization to be laser focused on one mission: to fund research that will accelerate prevention, treatment, and a cure for this disease. CureAlz aspires to empower great scientific talent to make real breakthroughs, combining low bureaucracy with a culture of challenge and collaboration. To that end, we have supported more than $140 million in extraordinary research. All of this is possible thanks to our dedicated donors who share our drive and determination to end this disease and its terrible impact on patients, families, and communities. We are held accountable for this mission by our board of directors. They, with certain other dedicated supporters, cover all of our operational overhead. That generosity means our wonderful donors know that their gift goes 100% to support research. The science we fund is selected by our research leadership group, comprised of experts across scientific specialties relevant to Alzheimer's disease. All of our scientific decision-making is done by active researchers who are themselves at the forefront of Alzheimer's discovery. In turn, our portfolio of scientific projects is assessed by our Research Strategy Council, whose members have demonstrated expertise in creating clinical value. Dr. Vince Groppi is a member of the CureAlz Research Strategy Council due to his long career translating academic ideas into patient treatments. Currently CEO and co-founder of Auricula Therapeutics, a startup biotech company developing drugs to protect hearing and balance from the effects of autotoxic drugs in aging, he was formerly the director for the Center of Discovery of New Medicines and director of the Center for Chemical Genomics at the University of Michigan, a consultant for the NIH-NINDS Blueprint Therapeutic Network, and is currently a member of the NIH-NINDS Translational Review Panel. He has held numerous other positions in industry, including leading teams that have advanced 9 compounds into clinical development. He holds a doctorate from the Robert Wood Johnson Medical School and was a postdoc and assistant professor at the University of California, San Francisco. Dr. Groppi, thank you so much for being here today to share your perspectives on the roles of venture capital and philanthropy in achieving meaningful treatments for Alzheimer's patients.
Thank you, Meg. Hello, everyone. Although I can't see you, I'm hoping that in other times we can actually meet in person. So my agenda today, to get everyone at the same starting point, is to very briefly give an overview of what drug discovery and clinical development is, go through the hallmarks of Alzheimer's disease, give a short overview of what the clinical pipeline looks like, and then provide 2 examples of standout projects that are funded in part by the Cure Alzheimer's Fund committee and show how they are adding to the gaps that are needed to actually, bring disease-modifying therapy to Alzheimer's patients. I'll talk at the end about what I call Cure Alzheimer's disease secret sauce, what makes them special, and then I'll return to academic drug discovery and go through their strengths and weaknesses, and I'm sure that will be the starting point to address questions about how to make things go even faster. So the first slide is, okay. First slide is really to introduce me to you a little more fully, to give you a sense of who I am so that when I talk, you'll have an idea of the frame of reference that I'm using. So I was part of three large pharmaceutical companies for 23 years, Upjohn, Pharmacia, and then Pfizer. During that time, I led cross-functional teams that put five new chemical entities that would be new drugs, new small molecule drugs, into clinical development for Alzheimer's, schizophrenia, bipolar, and unipolar depression. I actually started the bipolar drug discovery effort at Pfizer, which is something I'm very proud of. Along the way, and this was not anticipated, I was actually a tool builder too. And early in my career, I needed a tool that didn't exist that was able to measure essentially cell responses very fast so that you could do high throughput screening on a target, not using simple static binding assays, but actually doing functional pharmacology with cell models that would be similar to the disease themselves. The name I gave to the technology was Flipper, and really what it did for the first time in 1992 was able to read an entire 96 well plate. It's sort of the plate that you use when you do high throughput screening, 96, ultimately 384 and higher, but at the same time, and that basically unlocked the functional pharmacology of ion channels and GPCRs. Being a member of the Upjohn company at the time, all the inventions that I did was owned by Upjohn, but the sales of the technology has continued, and the instrument sales are over 1 billion. So it was a very successful idea. It's picked up by almost all drug companies and academics that do this sort of work, and it gives you an example of how I don't let little things like I don't have the right tool stop me when something is needed. When Pfizer closed its shop in Ann Arbor, Michigan, the engineering team that I built Flipper with called me up, and they had a new name at the time, but it was the same group. They were called SN Bioscience, and they wanted to develop another technology. It was essentially a slower version of Flipper. So where Flipper read cell events very fast and looked for rapid signaling that you would get from an ion channel or some molecule that causes fast events. There are many events in biology that are slow that you can only see basically with time-lapse imaging. And so what the SN Bioscience, as CSO of SN Bioscience, what I did in that case for about 7 years was to develop a technology that could quantify essentially time-lapse imaging. And why would you need time-lapse imaging in biology? Well, you need it to quantify things like the slow development of organs such as kidney or brain or heart that you can now do in a dish. An instrument that could essentially a microscope-based instrument that could do that with live cells, unlocks the potential of seeing how to intervene both positively and negatively. And the applications that my team developed were ones that in fact quantified organogenesis. They quantified neuronal, measuring functional neuronal activity, angiogenesis, that being blood vessels, cell migration, things like simple cell migration, cell invasion, cell growth, and cell death. And it was very successful, like Flipper was. And in this case, Essen was acquired by a German company called Sartorius for $320 million. That gave me the opportunity to go and think about what else I wanted to do. And actually while I was at Essen, I joined the NIH because, if you can imagine going from a company as big as Pfizer with over 100,000 employees to a small company like Essen at the time with only 10 employees, you basically went down 3 orders of magnitude in the number of people you interacted with. And so becoming part of, the translational medicine panels at NIH helped me keep my finger on the pulse. And as CSO, I thought that was one of my biggest jobs. And that led me to become a consultant of what I talk about a lot, which is the Blueprint Therapeutic Network. You may never have heard of that, but it's a very special part of the NIH, and it grew out of when the director of NIH named Francis Collins, when he was looking at what the translational grants were doing, what he found is that great research was being done, but it hardly ever got past what I call the bookshelf. So it would go from the bench to the bookshelf, but it wasn't going from the bookshelf to the bedside. And why was that? Well, it's basically because academic drug discovery, it's hard to get things, out of the, hypothesis testing stage into the linear stage of doing essentially the work needed to go from a hypothesis to a small molecule to an improved small molecule to a clinical candidate and ultimately a clinical trial. And the Blueprint Therapeutic Network proposed a way of doing it. And I was the consultant for putting that together and became a member of it. And the way it works is essentially academics with ideas and data for a drug discovery project would apply for funding. And when they got funding, the academic lab itself only got a small portion of the funding. So they got a small portion of the funding to do essentially the primary pharmacology and maybe some secondary tests. The rest of the funding was used by CROs. So in very much it worked like a virtual biotech did. So a small group of experts, and then you bolted on, the CROs you needed to do the work. So medicinal chemistry, PK, pharmacokinetics, safety, all of those were bolted on like a biotech would bolt on and used as for just-in-time research. It was really novel. NIH had never done it before. They brought a bunch of consultants like me in to do it, and we were used as co-leaders of these teams. They were called lead development teams. And in fact, I was the co-leader of 5 lead development teams. One was for this aminoglycoside induced hearing loss that Meg told you about. Another was for gamma secretase modulators that are used for Alzheimer's disease. They're embedded in a lot of what CureAlz has doing. I was co-leader of a PD, a phosphodiesterase 4 for improving the cognition of Alzheimer's disease, a Parkinson's project that was looking to block calcium overload in the substantia nigra, and a neurosteroid modulator for multiple psychiatric disorders. That's a long laundry list, but in a very short period of time, 4 of those 5 projects actually went into the clinic. So it was an incredibly successful project. And that model was one that you could use to unlock and accelerate projects coming out of academics. The Auricular Therapeutic LLC that was formed was in fact a spinoff of the Blueprint Biotherapeutic Program, and was built on science at University of Washington in Seattle and the Fred Hutchinson Cancer Center. We now have a CEO. I'm running the operation now, and we have a phase 2 ready drug to go in to prevent aminoglycoside-induced hearing loss, which is a dose-limiting side effect, and it's a first-class drug, and it will hopefully expand the use of the antibiotics for life-threatening illnesses. I then went also to the University of Michigan, where I took the model of Blueprint Therapeutic Network and basically made it fit for purpose at U of M. So I got the funding needed, built the infrastructure, and advanced 50 projects into, started and advanced 50 projects, and had a budget of about 1.5 million a year. And in those 50 projects, what we wound up doing, because as a person involved in a lot of business, you always like to know if what you're doing is successful. And one way of measuring success is looking at the return on the investment. And our return on the investment was about 9-fold. So for every dollar that the University of Michigan invested in the center, $9 were generated with downstream funding from grants, licensings, and collaborations. I think the crowning success was when Deerfield came in and committed up to 130 million to commercialize drugs coming out of the university. And a big part of that was because the medical school there, like a lot of medical schools, is just, it's just pregnant with possibilities to going forward. And the center that I had put together, which served the entire school, was essentially the launching pad. And Deerfield saw the advantage of that and are now partners in what's going on in Michigan. And lastly, as you know from my presentation here, I'm a member of the Research Strategy Council, where we help to identify and support the projects needed to essentially improve the standard of care of diseased patients. So that's probably a little longer than I intended, but it gives you a sense of who I am. In a nutshell, if you wanted to divide up basic science research from translational research. This is my summary slide to talk about that. And in basic science research, it's always PI driven. PI is a principal investigator. So there's a principal investigator that runs the lab. He or she is essentially the person responsible and accountable, and their career is dependent on the choices they make. Their product, by and large, is advancing scientific and medical knowledge. They get known for their novel scientific discoveries, and the more impactful those discoveries are, the more successful the PI will be. You can think of them as being an inch wide and a mile deep. So they're very narrow, but they're deep experts in their area. To the deepest extent, they know just about everything about a particular area. And it's often sub areas, like within Alzheimer's, it would be a sub area of Alzheimer's. For instance, the gamma secretase would be one. And, someone like Steve Wagner, who unfortunately recently died, was one of these people who was a deep expert in the way gamma secretase modulators could be used for Alzheimer's disease. They publish papers, they train students and postdocs, and they get funded largely from the government. If you then think across, well, in drug discovery, it doesn't really work like basic research, because drug discovery by its basic nature crosses boundaries. So you start out with usually a biology-driven or sometimes pharmacology-driven project, but it very quickly becomes a chemistry-driven project because, or if it's going to be a biologic, one that will involve, engineering, biology engineering project. But you go from an expertise usually of a team leader to an expertise of another team leader in a stage-dependent manner. And unlike, basic research, the goals of translational research is essentially understanding a disease and adding to improving the standard of care. So you start out knowing what the standard of care is. In reality, that means you work very closely with the clinicians involved in taking care of those patients, and you now look to improve the standard of care. It's milestone driven and not kind of open-ended with basic research. Milestone driven means that if you're trying to develop a drug for a particular indication, you basically put together a critical path milestone plan that goes after that drug. And you like to fail as fast as possible because, as I'll tell you in the next slide, many, if not most, drug discovery projects are not successful. So to conserve resources and time, you want to do the most difficult and challenging critical path questions as early as possible, so if there's a path forward or not. The team is broadly cross-functional. And, at the end of the day, you're going to create a drug, a device, or a diagnostic. Early funding also comes from agencies like the National Institutes of Health and the Department of Defense, but also major contributions are made from foundations, disease alliances, and private sectors more and more. So this is probably one of the most important slides I'm going to show because it takes you through what I think is the real world example of drug discovery. So drug discovery starts with a foundation of understanding the disease and using models that are translational. And what I mean by that is that when you build a model for drug discovery, that model has to be one in which if you get a positive or a negative result, it has direct consequences on understanding whether you have the ability to, treat the disease of interest or not. So the translational models are the foundation of it. And why this is a tortuous path is because you sometimes see these drug discovery and early development out as a linear event. It's not linear at all. It's almost always tortuous like this. And the reason why I have these two red arrows that you can see, one at the bottom and one at the top, is that in my opinion, both the translational models have to be built on human data. And there's three types of human data that it's built on. Usually it's human pharmacology, multi-omics data, or actual human genetics. And so the combined evidence from those disciplines are used to build a hypothesis of how to attack a disease. And that disease is one that you build a translational model. But if it's a new disease that's never been approached before, a new target for a disease that's never been approached before, how do you in fact know the model is translational? Well, you don't until you get into clinical development. So you do the best you can in early discovery, which is to set up the assays, late discovery, which is to tune the chemistry and make sure you have safety and the ability to get to the drug to the organ of interest. But it's not until you go into exploratory clinical development that you know whether your drug actually will have the ability to make a significant impact in patients' lives. And the reason why I have the model, a line coming from early development back to the clinical model, is that that's how you tune your clinical models. You tune your clinical models best. Your translational models are tuned by clinical data, and there's just no other way to do it right now. And, you can build a hypothesis from animal experiments, but animal experiments, by their definition, usually are not fully translational to disease. They often pick out some variants of the disease, but not the whole complete disease. And what's shown in the upper left is a study that I actually contributed to back in 2004. So in 2004, a set of companies, including one I worked for at the time, gave their data, their clinical trial data, to a third party, who then did an analysis about the success rate of drugs entering phase 1. So if you look at what's circled in 2004, the sad state of affairs was that if you started 100 projects in phase 1 drug discovery, you might get 10 drugs out the door at the end of the line. So there was about a 90% failure rate. And everyone in 2004 said, okay, we'll fix that. Well, this has not been fixed. And there's been multiple follow-up studies done subsequent to the one that was originally done in 2004. And the clinical trial failure rate remains in the 90% range. It's better in some versus others. Oncology is usually the lowest. Diseases that are better modeled by animal models, such as cardiovascular disease, are usually high. Infectious diseases are generally high. Infectious diseases, you kill a bacteria when you kill a bacteria, but it's often about biodistribution of the agent. So, but in general, this whole process is set up to essentially need clinical data in order to define how to advance the field. This is just an overview of what the approvals look like over the past 20 years. And, you can see by and large, on the orange bars are the small molecules. The blue parts of the bars are the biologics. So in 2020, it was, there were 40 new molecules approved and 13 biologics. And you can see it bounces around a little bit, but it's in the 20 to 30 to 40 range, and it has been that way for a while. And so that just gets you a sense of what's there. Now, if you back out from that, you can say, okay, if those are the ones that are approved, then there's 9 more that went into clinical trials that haven't been approved. And what's interesting about that is that those 9 that failed have enough safety data and PK data to actually make it into clinical trials. So when you start thinking about repurposing, people often think about repurposing the drugs that have been approved, but the repurposing you should also consider the drugs that entered phase 1 that never got out, because even though they didn't get out, they were probably safe enough to try. In most cases they they just didn't have the efficacy for the indication first gone after, but they might work for something else. So repurposing, which I'll get back to in a while, should be done with a set of drugs that include all drugs that have gone into clinical trials, and that amounts to about 30,000 drugs in that category. So here's one slide that gives you an overview of the major milestones in developing drugs for Alzheimer's disease. It was first identified, as probably most people know, by a German physician in 1906, and the physician Dr. Alzheimer's, and after which the disease was named, found profound memory loss of a patient, and it was one where the memory loss was continued over time. So it was a progressive memory loss. What's often not stated because it's a different field, but if you look at psychiatry at about the same time also identified schizophrenia. And schizophrenia was originally actually initially defined as a problem in cognition, just like Alzheimer's. The difference between this schizophrenia in those years and Alzheimer's was it was not progressive. So schizophrenia shows up early in usually a patient's life cycle, somewhere in the late teens, early 20s, but unlike Alzheimer's disease, doesn't progress with the same trajectory. In 1993, so I worked for Pfizer. As I mentioned, Pfizer had acquired Park Davis, so I worked at the old Park Davis site. There were people at the Park Davis site who remembered in 1993, I heard the story multiple times about, Park Davis had a drug at the time called Tacrin. Tacrin was developed to be an acetylcholinesterase inhibitor, and FDA actually came to Park Davis before 93 and asked them to bring the drug into clinical trials so that they could determine whether what was called at the time the cholinergic hypothesis had merit. So it was one of these nice interactions with FDA that brought a drug that wasn't a particularly good drug, but it proved a concept. And the concept was if you blocked acetylcholinesterase and raised the level of acetylcholine in a patient, you could have a mild effect on increasing a person's cognitive deficit in Alzheimer's disease. After Tacrin was shown to be effective, there was a fast follow-on with 3 other cholinesterase inhibitors, one called donepezil, one called rivastigmine, and the other is galantamine. They all work more or less the same way, which they inhibit acetylcholine, and the dominant drug that came out of this field of research is donepezil, which carries the trade name of Aricept. And most patients who have Alzheimer's disease at some time gets treated with Aricept. There's another drug that came several years later. It's called memantine. Memantine was an old drug that was originally designed to treat glutamate overload in, I think, stroke. It's a very weak, use-dependent blocker of a receptor, ion channel called the NMDA receptor, and it does a weak blockade of that. And it, in fact, also is approved for symptomatic relief of Alzheimer's. And then nothing really happened until last year. And so those were the drugs that are used to treat Alzheimer's. They're not very effective. They give small relief. In this country, most Alzheimer's patients will be on them because it's better than nothing, but there's no change in the course of the disease. There's usually just a small change in the improvement of cognition. The neurologists that I know that treat patients say that you basically buy a patient something like 6 to 12 months of additional, additional cognitive improvement. Last year, as probably most people know, aducanumab was approved as immunotherapy because it targets the amyloid plaques of brain. So it does a very effective job at removing one of the hallmarks that I'll tell you in a minute of Alzheimer's, the amyloid plaque deposit. And they received accelerated approval by FDA. Just this year, not too long ago, a couple of months ago, in fact, what the CMS proposed, that was the part of that Medicare will only cover the cost of the drug if you're in a clinical trial. So the price of the drug currently is $28,200 a year, as set by the company that launched the drug, which is Biogen. So I'm just showing you that of the drugs that are out there, there's some controversial disease progression relief, some symptomatic relief, but there's a long way to go before we have enough, we have a drug good enough that can actually make us feel good about treating Alzheimer's patients. The hallmarks of the disease from the very beginning are really three. There's more than this, but these are the three big ones that we'll return to a couple of times. One is the amyloid plaques themselves. These are deposits found outside the neurons. There are tangles of another protein called tau that's inside the neurons. And what comes along for part of the pathology is the neuroinflammation. So these are the hallmarks of the disease. They're currently, if you go into the clinical trial database, 126 drugs in clinical trials for Alzheimer's disease. Most of them target disease modification, looking at either the plaques, the tangles, or the neuroinflammations. Right now, there's about 38,000 participants, so an enormous number of participants in these trials, and they're largely run by the pharmaceutical companies. 49% of them are directly from the pharmaceutical companies, and another 14% are the pharmaceutical companies collaborating with other sponsors, usually academics. So that gives you the backdrop of drug discovery and Alzheimer's disease. The other major thing that I think is now very clear is that there are pre-symptomatic as well as symptomatic stages of the disease. So what you're looking at here is a series of biomarker changes that happen, as over the course of the disease. And if you look at what is the zero mark here, the zero mark sets when a neurologist can start picking up changes in memory. So this is essentially where the memory tests, the neuropsych tests that are given to Alzheimer's patients, where they can pick up changes in symptoms. But before that change, usually decades before, there's the beginning of the deposition of the amyloid plaques, the formation of, you can start finding the tau protein in the CSF. The tau pathology actually correlates very closely with the symptom formation. And what happens is the neurodegeneration, which you see here in blue, the neurodegeneration starts falling even before you see symptoms. And then there's the glucose metabolism is just a general way of looking at brain activity. Glucose metabolism starts falling. So to be successful, I think, historically, the way trials are run is they're run when you start to pick up symptoms, because you need some measure to determine whether your drug is effective. So you need to have memory loss in order to see if you're protecting against memory loss. But if you start treating at the time where you're picking up symptoms, it may be too late. And that's what this is meant to show. So that, if you start thinking about where you'd like to treat well before you start seeing any of the biomarker pathology that is meant that most of us think are causally related to the disease. And so to do a prevention trial, is a high hurdle, because you're now taking otherwise healthy people. You're taking otherwise healthy people and you're using them to treat, something, to something at risk of happening. So it's not easy to do, but it's essentially the way the field is going. And so that's what this slide basically said, is that focusing on the molecular pathways rather than symptoms themselves has nurtured the need to discover and develop a better biomarker. And I'll go through biomarkers in a minute. So you can detect AD at the pre-symptomatic stages. And that detection then allows for early intervention. And the ultimate goal is to treat pre-symptomatically before the drugs are before the disease has worsened, so that you could have a better chance of changing the course of the disease. So what's a biomarker? A biomarker, is essentially a biological test that can objectively be used as an indicator of disease progression. So everyone knows biomarkers because everyone in the audience have probably had their cholesterol tested. So cholesterol is used as a biomarker for atherosclerosis and cardiovascular disease. So if we all have our cholesterol below 200, we're pretty happy. So what we want essentially is a biomarker like cholesterol is for atherosclerosis. We want a biomarker that's like that for for Alzheimer's disease, because just like we can use cholesterol, decreasing our cholesterol levels to help us feel as though we're protecting ourselves against cardiovascular disease, if we could have biomarkers that are tied to Alzheimer's pathology, and we can use those, and those are easy to use blood biomarkers in particular, then we can use those for early interventions. The criteria of a biomarker is pretty simple. It has to be highly specific for the disease. So in other words, the on target versus off target has to be high, and it has to have a high sensitivity, so that with a small amount of blood draw, you can actually detect it, so you don't have a false negative result. So with that as a backdrop, one of the very exciting things that have happened at Cure Alzheimer's since I've been part of it is the work of Randy Bateman. So Randy Bateman is a professor of neurology at the at the Washington University School of Medicine, WashU, and he's given me permission to show these slides. So I just want you to know, if you wanted to know more about this, Randy and his colleague, Dave Holtzman, are the world's experts on it, and it's been a pleasure to work with them. And so I'm now showing, essentially a small snapshot of their data, and their data looks like this. So what they did is they developed the first FDA-approved blood test for amyloid pathology. And what you see on the left is essentially a curve showing the sensitivity and specificity when they start looking at measuring if they look at the ratio of A beta 42, which is one form of amyloid protein that you can detect in the blood, versus A beta 40. And when they look at that ratio and couple it to one of the other biomarkers for Alzheimer's disease, which is a protein called ApoE4. So with the ratio of A beta 42 and 40 at ApoE4, they have a 90% rate. They have a 90% predictive value. So they formed a company. So all this work was supported in part by the NIH and in part by Curals, and it led to essentially this very exciting blood biomarker. And they formed a company called C2N, and C2N now has got an FDA-approved drug that can actually do this measurement. They have an automated system that if you're a neurologist, you can send your blood samples to C2N. They'll do the analysis and send you back the data. Now, what's really important to state is that the test per se doesn't diagnose Alzheimer's. It does not do that. What it does is it gives you a blood test that is highly correlated with what you would see if you did a brain scan of a patient that had amyloid plaques. Now, amyloid plaques is one of the contributing factors to Alzheimer's disease. So what you have here, in my opinion, is one of the first tests that's going to be used. It alone is not sufficient, but it's going to be one of the tests for which others will be added, so that we can eventually get to a point in Alzheimer's disease. So we have the equivalent of what I said earlier was needed, which is the cholesterol version of Alzheimer's disease. So this is a major step forward. It's very exciting. I'm sure there's going to be lots of tests that will follow this, and this is the first one, though. And both Randy and Dave are member of Cure's Alzheimer's, very active members, and their work is proceeding as well. The second example that I'm going to show comes from Rudi Tanzi's lab, Rudi Tanzi at Harvard. He's essentially one of the science leaders at Cure Alzheimer's. He's also a very infectious guy, in the sense that he wants, I think if he could have it his way, he'd have everyone working on Alzheimer's disease. That's how passionate he is. He certainly got me involved. And so what I'd like to do is just show you one of them. So Rudi is multifaceted. This is only a part of what he's doing, but I think it's a significant part. And he's basically developed a model of Alzheimer's disease from cells and culture. So he calls it the Alzheimer's disease in a dish model. And what he does in a very complex but reproducible way is he puts together, the cell components that are needed to essentially model Alzheimer's disease pathology in cells and culture. And when he does that, he can model the three hallmark pathologies, that being the beta amyloid pathology, the tau pathology, and the neuroinflammation pathology. And this is all published work that he's had for a while, and the models have gotten better and better with time. It sits in a 96 well plate. This is the sort of plate I told you that I originally used, the flipper technology to read and the AcuSec technology to read. Well, Rudi's lab is using their own tools to make the same sort of reading over a period of days and weeks. So with being able to quantitate amyloid, tau tangles, and neuroinflammation, he can now start screening drugs to see if he can find any new ways of treating them, and being impatient and wanting to go forward as quickly as possible. He started with drugs that are already out there for other diseases. So these are drugs that are on the market, as well as natural products that have a very short launching pad to get to clinical trials. And that was his collection of drugs that he started with. And the profile of activity that he has right now is 10 of these have been identified that reduces tau, as well as they increase the glial uptake. 9 reduce both phospho-tau and tau, and in a different type of neuroinflammation model, one driven by TNF, and 6 are both tau and A beta. So he's bucketed these in different categories, and he's now got a group that's called Act Fast. They're trying to parse out the best of these and see how quickly we can actually enter a clinical trial with some of the discoveries he made, because it goes back to the beginning of so what we have right here. So if you think about what I said initially, this is Rudi's disease in a dish model. He's got a very fast track approach to finding activity. He now needs to know whether that activity is going to impact the disease in the clinic. And the way to do that will be using a fast biomarker study to see if the biomarkers for the hallmarks that are affected in the dish are also affected in the clinic. So that's essentially the fast track approach to finding new therapies that will be effective, and then finding which ones are translational and which ones aren't. So, right. So I think I just told you all of this, right? So I'm going to end with two last slides. And the first slide is just to talk about what I call the secret sauce of the Cure Alzheimer's Fund. I think for those of us who've been in organizations that are successful versus unsuccessful, it starts with the people. So, so if you have the right people, in the right setting, magical things can happen. And I think that's really what I see the Cure Alzheimer's Fund secret sauce. It's getting the best researchers that want to work together on a project. And the structure of it is an open and borderless scientific and medical community that are, in fact, experts in all aspects of Alzheimer's disease. And the culture of collaboration and accountability is baked into the cake. So that's part of the DNA of the group is to collaborate, because working alone just won't be effective at treating a disease as complicated as Alzheimer's. Everyone knows that. There's very strong and very transparent leadership by several of the people I've already mentioned, Rudi Tanzi being one of them, Dave Holtzman being a second, and Bob Vassar at Northwestern being a third. They're all open folks that will share their data freely and take constructive criticism well. They work in concert with the board, as well as the Research Strategy Council that I sit on, to make sure that the best high-risk, high-reward projects are identified and supported. There's a deep commitment of all the members, as long as I've been on, a part of the organization. there's a deep commitment, meaning that people freely dedicate their time to work on new things that are needed, and there's a feeling that everyone has that we want to use our knowledge that we generate to catalyze the discovery and development of new therapies and diagnostic tools. I showed the Randy Bateman's diagnostic test as one of them, and there's others as well. I think the organization's role is pretty clear. It wants to foster and de-risk the promising projects that would not get funded easily by NIH, because NIH, although the National Institutes of Health, although they have an enormous budget, are still risk adverse. I sit on those committees, and I know that only a fraction of the good grants that come through actually get funded. And what Cure Alzheimer's does is it essentially allows these high-risk, high-reward projects to generate a data set that now makes them competitive when they go for funding from something like the National Institute of Aging, which has an Alzheimer's budget over $3 billion, which is enormous. And there's a minimized bureaucracy and reporting, and it wants to foster maximum impact. The last slide is just to return to academic drug discovery, and I'm just kind of highlighting what I showed you in the graphic earlier, which is that the advantages of academic drug discovery are pretty clear. They have a deep expertise in Alzheimer's disease, and they have direct access to patients and a thorough understanding of their medical needs. They have sophisticated assays, reagents, and models that are improving over time. Rudi Tanzi is being one of them. Another thing for the venture community to understand is that all of the funding they get, all the funding that's gotten from National Institutes of Health is totally non-diluting. When I was at Pfizer, and when Pfizer wanted to spend $5 billion a year on its research budget, if $1 billion of that was in basic science, that came directly from shareholder value, right? So and so this is a way of essentially generating the hypothesis without diluting capital in any sense of the way. And another thing that I learned being at Michigan is that that if you have a basic science individual and you start to convince that person that there's a translational advantage for their research, they can actually get two sources of funding. They can continue with their basic science funding. In fact, they'll probably be in a better shape to get it because they now have a real effort doing translational medicine. So that parallel track can be part of what an academic uses to keep their lab growing. The disadvantages, there's several disadvantages, of course. The first is, as I said earlier, that most academic drug discovery are unfamiliar with true cross-functional team-based research. They do team-based research, but it's really research coupled in their own silo. So if they're immunologists, they're talking to other immunologists. But the cross-functional basis that I'm talking about, working with chemistry and safety and regulatory, that doesn't exist in academics by and large, unless you have a center like the one I created at Michigan. They're also very unfamiliar with linear, milestone-driven, fail-fast research, and that's the core of it. They're used to following the best science, and that science can often take tacks and does in ways that discover new things. But in drug discovery, you basically have a Gantt chart where you want to go, and you're either going to get there or not. And most academics are unfamiliar with that. And they're also unfamiliar with using contract research organizations to address critical path needs that are outside of their expertise. Now, all of those can be fixed by an organizational change like the Blueprint Therapeutic Network, but that's means changing the culture too. So with that, Meg, I think that's my that's the core of what I wanted to get across.
Great. Thank you so much, Vince. This is fascinating, and I have lots of questions I can pose to you, but I do also want to encourage our audience to do the same. If you type your questions into the Q&A, I can pose them to Vince and get his responses. In the meantime, I will go ahead and get us started and say, you've talked a lot about kind of the great things that academic researchers can do and the need to then think differently for commercialization. How do we bridge that gap? How do we make sure that the two sides are speaking the same language and giving each other the right information to be compelling? How do we get academics and VCs to partner well?
With academic labs, it's no different than any other situation, which is an assistant professor or an associate professor in academics are going to do what they need to do to be rewarded. So the reward system of universities and the reward system of the NIH is one that you just have to, you have to work within those boundaries. And I think the way I've seen it work best is to say, what can we do that doesn't, so the way a venture capital would work best with an academic, in my opinion, is you put together essentially a structure such that the academic doesn't have to spend any more time on the translational project than that person would essentially advising a student or a postdoc in their lab. So what you'll never do is you're never going to get them to do what I do for a living. But what you can do is get them to see the advantage of what I do for a living as something that they can do, but they need resources to do it. So essentially what you have to set up is a structure that gives them access to Is that clear?
You froze a little bit on my end. I'm not sure whether that was me or you, so I apologize if it was me.
Oh,
But it said you were saying that you have to give them access to a structure, and it sounds like, kind of let the academics stay in that creative space, but make sure that people with really the ability and the tools to focus and carry the ball forward also have room in the ecosystem. Is that a fair description?
That's exactly what I want. So because I think an academic who's good at what they're doing will want to stay at what they're doing mostly, but you can carve out part of their time, 10%, 20%, something like that, their time, as much time as they would use to direct a project in their lab. And what you have to build in with that is essentially the infrastructure needed for them to advance the project. So at Michigan, what I did at Michigan, which is very similar to what virtual drug discoveries companies do, is you bring in core laboratories. So there's a bunch of experts that have cores that can do the different components that are needed. There's a medicinal chemistry core, there's a protein engineering core, there's a structural biology core, there's a PK, a pharmacokinetic core, there's a safety core. You could either do that with core labs within a university setting, or there are contract research organizations that do that very well. But you need to be able to bolt on the downstream resources that currently don't exist and give them, essentially an expert to work with to help navigate all this, because you don't want on-the-job training. That's too painful.
And how do you help the academics answer the questions that any VC will want to pose? And how do you make sure that the VCs are seeing the potential in the ideas that the academics are looking at in a non-commercial, much more let's solve a scientific mystery space?
Well, there's all different ways that you can structure these interactions. One way that I favor is that if it's an early project, very early project, so if you're at the hypothesis generating stage and it's one that can be translational, but you don't have NIH funding to do that, and there's mutual interest from from someone in the venture capital world, then one of the ways that you can talk about essentially pre-seed funding, which this is that the pre-seed funding gives access, gives the VC access to the lab doing the work. So access equals influence almost always. And so that's how you essentially get the conversation going. And then what you get for the pre-seed funding is essentially something the equivalent of first rights of refusal. So you're there, you're partnering with what the opportunity is. but you don't have an equity stake in it at this point. You just have influence. And then if something were to come at it, you're the first person they talk to essentially make terms. And so the way one of the Japanese drug companies did it at Rockefeller is they essentially pre-cooked what the deal would look like if it went forward. So it blocked out the downstream negotiations, which sometimes takes years. So when you put this together with different, institutions, you can imagine what, the paths will be. And if you have those contracts already set up, so you've spelled out what a successful partnership looks like, and if that's in place, then that's one of the ways to move.
So I can't tell if I'm freezing or you are, so I apologize.
Oh, maybe it's me. I'm sorry.
Yeah, it might be me. But let me ask you a different question if you can hear me.
I can hear you. I can't hear you. Is it me? Is that what that means?
I think it was you, but you're back. If you can hear me, let me take us in a slightly different direction with a question about clinical trial failures. Um, One of the explanations that we frequently hear with why a clinical trial has failed is that, oh, there was some important fact about Alzheimer's we didn't know yet. Does that mean that we're rushing into trials too quickly? Are we asking people to put capital at risk before we really know enough to make those risks financially viable? Is that fair to patients? Should we be just focusing on basic research for a while longer before we go into trials?
Well, I'll go back. I heard most of that, and I'm sorry my connection's unstable. I'll go back and say that the only way we're ever going to advance the field is with clinical data, and so I think that, the more clinical data we generate with different targets in mind, the more we're going to understand what combination therapy, because it's almost certainly going to be combination therapy that we need in patient-specific manner. So having a panel of biomarkers to define the Alzheimer's disease phenotype in patients, because it'll be different patient to patient, and then understanding what drugs are able to intervene with the phenotype. Think of it as a fingerprint. So you have a fingerprint based on your biomarkers, and based on your fingerprint, you have now a collection of compounds that you're going to use to treat, your pathology, to reverse it back to where it is. And if it were cardiovascular, that most people understand, imagine you have a high blood pressure and a poor heart rate, right? So you want to get your heart rate, you want to get your heart back. You want to get your, by essentially increasing blood flow, you want to get your blood pressure down, and maybe you have some sort of kidney involvement. So just understanding the key features of the pathology is you come in with the different drug tools to intervene, and that changes with time as well. So, but that all gets back to clinical trials, Meg. I don't think you're ever going to get past testing the hypothesis clinically, and you want to do it as quickly as possible and as cheaply as possible, and always safe.
So how do we make clinical trials in Alzheimer's disease faster and cheaper than they are right now?
Okay.
Are there tools that we're waiting for? How could we do
I think
better?
there are tools. I think the tools are these biomarkers that are coming down. So right now, the biomarkers that are being used, to type the disease are all imaging biomarkers. These are ones where you image plaque, where you image tau, and these are very clunky. Thing. I mean, they're very efficient, but you'll never going to run a trial with an imaging endpoint. It's too costly and not quantitative enough. So you want blood biomarkers, just like what Randy and Dave have, that correlate well with the pathology at hand. So it's getting better blood biomarkers that correlate with the disease pathology, and then using the change in those biomarkers to determine whether the drugs are moving in the right direction. And if they are, then that's when you do your trial based on, based on symptoms relief, which would almost be the neuropsych test.
So, people within the Alzheimer's field are feeling pretty optimistic, I would say. There's the first drug that's been approved that some people believe is truly disease modifying. We have a blood test for the first time. Our imaging tools for amyloid and tau and even neuroinflammation are getting better. But it's also a pretty insular field. You have experience in a lot of different diseases. Do you think Alzheimer's is really at almost a threshold moment, or is this kind of more just regular progress? We'll see how it goes.
I think it's not a threshold moment. I think it's not a threshold moment. I think there are many diseases that have complex natures. And I think, being a neuropharmacologist, the ones that come to mind are diseases like schizophrenia and bipolar, but really you could probably put diabetes in the same boat. And I think what we're going to do is the hard, heavy lifting that's needed to, what I said in the beginning, that we do at Cure Alzheimer's. We understand the molecular basis of the pathology, and based on that understanding, we generate tools, those being drugs, that will reverse that will stop the pathology. And then those are the tools that we're going to now go back and test in the clinic. But do I think it's going to be fast? I don't think it's going to be fast, no.
Well, I know we're over time, but if I can ask for your forbearance, we do have one more question I'd love to be able to pose to you.
Yeah, of course.
And that is, to what extent have the failures of phase 3 clinical trials resulted from poor patient selection, including the use of later stage patients? Is it practical to enroll a higher percentage of pre-symptomatic patients? As your graph suggests, that may be critical to success.
Yeah, so one of the things, that's a very important question. And the reason why historically pre-symptomatic patients haven't been enrolled is because there's been a poor correlation between the conversion of pre-symptomatic patients to actually Alzheimer's. So if you think about it as a cohort, mild to moderate, maybe, what's called early MCI, mild cognitive impairment. So historically, trials have been run with patients that have some form of symptomology. And the reason for that is if you go, when there is no symptomology, you then have to ask the longitudinal question. So how many of those patients that you select actually go into Alzheimer's, and how long will that take? Well, if it's only 50% and it takes them 10 years, that's not practical for a clinical trial. And so that's why I think what's needed are the diagnostic tools that do a better job at identifying the pre-symptomatic patients. We just don't have that identification completely nailed down. But if I'm optimistic about anything, Meg, I'm optimistic about getting those, because there's a lot of good people working on them.
Well, it's nice to end with some optimism. And I do know that we're past the hour. So I want to thank you so much, Vince. This is, as always, such a pleasure to hear from you. I want to thank our audience, our founders, and our hosts for this event. I encourage anyone with follow-on questions to reach out to us, and we will get an answer from Vince. And I hope everybody has a great rest of the day. Thank you so much.
Bye-bye.